Hi, I'm Selim Kalayci, and I will be presenting our study titled ProNetView-ccRCC, a web-based portal to interactively explore clear cell renal cell carcinoma proteogenomics networks. Other researchers involved in this study are Francisca Petralia, Pei Wang, and Zeynep Gümüş, and we are all located at ICON Institute for Data Science and Genomic Technology and Department of Genetics and Genomic Sciences at ICON School of Medicine at Mount Sinai. National Cancer Institute's Clinical Proteomics Tumor Analysis Consortium, CPTAC, has been performing comprehensive proteogenomic characterizations of several types of cancers. And to identify drivers of cancer progression from these massive multi-omic CPTAC datasets, co-expression patterns between protein pairs are being characterized, which can easily be represented as networks. Furthermore, these CPTAC networks increasingly borrow information from other types of datasets generated on the same tumor samples, which result in large and complex networks that are challenging to communicate visually. To facilitate better navigation of the resulting high-dimensional networks, we developed ProNetView. In a nutshell, ProNetView is a user-friendly open-access web interface that enables interactive exploration of CPTAC networks to help translate them into biological insights and clinical applications. Now I would like to briefly explain why do we need such a tool in the first place, why existing tools such as Cytoscape are not suitable for our purposes. Even though existing tools such as Cytoscape has advanced and mature capabilities, they are primarily designed for network visualization rather than network exploration. Whereas ProNetView has been primarily designed for interactive network exploration. Another main distinct feature of ProNetView is that it is designed and customized specifically for CPTAC dataset and user community needs. Accordingly, we have incorporated multiple CPTAC-specific clinical and other metadata within the co-expression networks. Another criteria that is important for our purposes is the ease of access and use for the tool. For that purpose, we developed ProNetView as a web-based tool which runs on any modern standard web browser without any need to install additional plugins or libraries. Last but not least, ProNetView provides three-dimensional network exploration capability, which makes it easier to explore large and complex networks. Now I would like to briefly explain the study and dataset for which we deployed our initial version of ProNetView. Specifically, this CPTAC study was related to proteogenomics characterization of clear cell renal cell carcinoma, ccRCC, and was published in Cell in 2019. As part of this study, an integrated proteogenomic analysis was performed for 103 ccRCC tumors. Similarly, co-expression network was built using phosphopeptide data from these samples. Phosphopeptide data included more than 20,000 phosphopeptides mapping to around 5,800 unique genes. A random forest-based model was utilized in order to capture co-expression pattern of phosphopeptide level data at the gene level. Basically, two genes were connected if one or more phosphopeptides mapping to the genes were highly associated to each other. The estimated co-expression network involved around 3,600 genes connected by more than 11,000 edges. Also, based on the network topology, we identified groups of highly connected genes. An interesting finding that was highlighted in the article was that the cell cycle and angiogenesis enriched modules were captured only by phosphoproteomics data. Another interesting study was that the angiogenesis enriched module, which you can see on the right, was inversely correlated with BAP1 and chromosome 14 loss, as well as it was associated with lower grade. Now getting back to ProNetView, we have employed ProNetView, which we accordingly name as ProNetView-ccRCC from now on, for two types of networks within this study. First one is the main ccRCC tumor phosphoproteomics network. For this main network, users can interactively explore all modules and they can query gene level information. Second type of networks are the module-specific networks that are derived from the main network. For the module networks, users can perform deeper queries of gene level information. In addition, users can explore pathways that are enriched within the module. And finally, users can perform phenotype level queries. Now I will try to quickly introduce these features in the next couple of slides. But of course you can navigate to this URL and explore it in more detail yourselves. In this slide, you see the landing page for ProNetView-ccRCC. The main figure, the main graphics is the three-dimensional network for ccRCC tumor phosphoproteomics network, and you can rotate this network as well as zoom in and out of this network in 3D. On the right hand side, you see the functional modules, and also below that you see a search box. And using that search box, you can search for a gene of interest, for example, ERG. When you search this gene, it will be highlighted in the network, as you see, in red color. Also, associated metadata, such as connected genes as well as clinical variables, are popped up below the search box. For the module-specific networks, you can explore enriched pathways using this interactive table. You can click on a pathway name, and the genes associated with that pathway are provided as a text box. Also, they are highlighted in the visual in red color. Also, you can perform phenotype searches in the module networks. For that, you can specify a phenotype of your interest, such as grade, and you can specify an FTR cutoff, such as 0.2. And then the matching results are again provided as text box, and also they are highlighted in the network in red color. ProNetView is an ongoing effort. Currently, we are incorporating other SPTEC networks across other cancer types, data types, and tissue types. Similarly, we will be adding clinical and metadata associated with each network. We will also improve the user interface and add new functionalities based on the user feedback. We will also employ analytic tools to measure user behavior and statistics and upgrade our tool based on the user needs. Finally, I would like to let you know that our publication is available. It has been recently published by Proteomics Journal, and you can see more details of this study in that publication. And also, I would like to acknowledge the grant support by NIH NCI's SPTEC grant. And finally, I would like to let you know that we allow every kind of feedback, any questions, any feedback, any feature requests. Please let us know from these email addresses. And I thank you.